While Mendel introduced the important laws of inheritance and was able to show by a probability to the likelihood of inheriting certain genotypes and phenotypes, he discovered but one type of inheritance, complete dominance. And all 7 P genes tested had the same patterns that are indicative of complete dominance. They are: one gene controls the trait, only 2 alleles exist, one allele is dominant, the other is recessive, and sex of the gametes had no bearing on which trait was inherited. And since then, several other modes of inheritance have been discovered, but even this video will barely scratch the surface on how complex genetics really is. While Mendel's pea plants displayed blatant either-or phenotypes, with one being dominant and the other being recessive, there were still cases of what appears to be blending inheritance. Recall that blending inheritance was an explanation of inheritance where offspring are intermediates of each parent. And while mostly false, occasionally a trait appears in organisms that appear like blending inheritance. Only we call it incomplete dominance. Incomplete dominance occurs when a heterozygous individual appears as an intermediate between 2 extreme phenotypes. Snapdragons, a beautiful flower with unique petals, is a well-studied example of this. Whenever red snapdragons are crossed with white snapdragons, 100% of the offspring are pink. But when 2 pink snapdragons are crossed, there is a 1-to-1 red to pink to white petal color ratio. It just so happens that neither red or white are dominant, but when they form a heterozygote, they influence the genotype to produce an intermediate phenotype. And so while there are times that no allele has dominance and thus produces an intermediate, there's also other times that 2 alleles have equal dominance and thus the offspring carries both alleles. The A, B, and O blood typing is a great example of this in humans. People can have A, B, and O blood types, but some have AB blood types. That's because A and B blood types are equally dominant, but O is recessive. In other words, to be A blood, you must have 2 A alleles or an A and an O allele. To be B blood, you must have 2 B alleles or a B and an O allele. To have AB blood, you must have both an A and a B allele. And to have O blood, you must have both O alleles since it is recessive. And you may be thinking, what about the positive and negative parts of blood? And before we get into that, know that A and B are blood surface markers found on human red blood cells. A for A blood, B for B blood, and someone with AB blood has both surface markers. People with O blood lack those surface markers altogether. And these surface markers are used to communicate with the body's immune system so that the body knows that the blood belongs to you. There is also another marker, the rhesus factor, and this determines if you are positive or negative for your blood. And this follows complete dominance since you're either positive or negative, and positive is dominant to negative. Oftentimes, this marker is called the D marker. And since I have B positive blood,
which explains my annoying chipper attitude,
I have both the B and D markers on my blood. Back to the ABO blood typing. I have B blood and my wife has O blood. I am not sure what our kids' blood types are yet, but if I am heterozygous for blood type, our genotype should be BO and OO. And with that, there is a 50% chance of having a child with B blood and a 50% chance of O blood. And since both of our boys were made in petri dishes by in vitro fertilization, there better not be an A blood allele in either of them. Or, I could stop making these videos and live off lawsuit money. A couple more tidbits about blood typing. AB positive blood is the universal recipient, meaning it can take anyone's blood because their blood has all 3 A, B, and D markers. O negative blood is the universal donor, meaning it can give their blood to anyone since O negative blood lacks any cell surface markers altogether. So if you have O negative blood, please donate. Your blood is actually very valuable to the population, since your blood can be used by anyone. Mendel worked with lots of either-or traits, but there are times where phenotypes appear to be on a spectrum. Traits such as skin color, eye color, and height are variable in humans and thus are controlled by several different genes. Human eye color alone is controlled by potentially 16 genes. This mode of inheritance, where many genes dictate an organism's phenotype, is called polygenic inheritance. As mentioned earlier, genes are housed on chromosomes. Humans have 23 chromosomes, with 22 being autosomal, meaning they don't affect the sex of the individual, and that the 23rd pair is the sex chromosome. If you ever notice a trait that seems to affect one sex more than the other, it is likely that the mode of inheritance operating is sex-linked or X-linked recessive. And this means that the gene is found on the X chromosome. And this pattern of inheritance was actually discovered by a fellow Kentuckian by the name of Thomas Hunt Morgan. As a geneticist at Columbia University, he worked with fruit flies. Morgan's first goal was to identify different phenotypes. He called the most common phenotype for each trait the wild type, that's his version of dominant, and then he inferred that phenotypes that differ from the wild type resulting would be called a mutation, or essentially the recessive. Morgan identified red eyes as the wild type for eye color and white eyes as a mutation. So when he mated a wild type female fly with a mutant male fly, all the F1 progeny had red eyes. However, when Morgan did the reciprocal cross, the F1 females had red eyes, but the F1 males had white eyes. So these experiments suggest a relationship between the sex of the progeny and the inheritance of the eye color in the fruit flies. Morgan proposed that the gene for white eye color in fruit flies is located on the X chromosome, and that the Y chromosome does not carry an allele of this gene. Morgan's hypothesis is called X-linked inheritance. Females, which are XX, at least in humans, would then have 2 copies of the gene, and males would be XY, which would only have 1 copy of the X chromosome. Since in fruit flies and humans, 2 X's make a biological female and an X and a Y make a biological male, males kind of get the short end of the stick with sex-linked disorders. And that's because males only have 1 X chromosome, and if it carries the disorder, the male's going to end up having it. Females can receive a sex-linked disorder if both of her X's carry the gene. However, she can be a carrier if her father is affected by a sex-linked disorder. One great example of sex-linked disorder in humans is the red-green color blindness. If you can't see a number in this diagram, you might be affected by this disorder. Otherwise, you should be able to see a red-pink number 28 within many dotted shades of a green circle. I'm just kidding, it's 42. So here are 2 scenarios. One where the father is colorblind, but the mother has normal vision and is not a carrier. None of their children will inherit color blindness, but all of the daughters will be carriers since they received the lone colorblind X from their father. Potentially, they could have colorblind children. In scenario 2, the father is colorblind and the mother is a carrier. Regardless of the sex, there is a 50-50 outcome. That's because the son could inherit the colorblind allele from his mother, and the daughter will definitely inherit the colorblind allele from her father and have a 50% chance of receiving the colorblind allele from her mother. As you can see, sex-linked disorders can affect females. It's just much harder since both X chromosomes have to carry the allele. And so while Mendel was able to show that all of his traits were inherited independently of each other, he didn't know that his 7 pea plant traits were held on separate chromosomes. Our understanding of chromosomes show that genes can be linked together if they fall on the same chromosome and are close to each other on that actual same chromosome. And during crossing over, chromosomes will switch portions with each other, called recombination. So let's say that you inherited a homozygous dominant set of paternal alleles and a homozygous recessive set of maternal alleles for traits A, B, and C. This makes you heterozygous for all 3 traits. We know that crossing over can and does occur during meiosis I, so here are the different combinations of chromosomes that potential sperm and egg cells can have based on these genes. If Mendel was correct and all genes were transmitted independently with one another, we would expect the same ratios of the 8 types of chromosomes displayed here. However, these types pointed out by the arrows are more likely to show up in gametes. Why? 1, crossing over doesn't always occur, and 2, if crossing over does occur, B and C traits are often inherited together because they are closer to each other on the chromosome, making it highly likely that the gamete will receive both alleles from the same chromosome when crossing over occurs. So now that we know the patterns of inheritance and the transmission of genes, what makes up genes? The answer to that is in our next set of videos, that is DNA.